Hi, and thank you for joining the tutorial. This program is about creating and planning your plate-based assay experiments and organizing your data. So let's start right away with a quick example. Okay, so first I am defining the experimental parameters for the experiment. For the example, I will show the use case of our company. So first we have the temperature in Celsius degrees. Then we have a plastic type, which doesn't have a unit. And we want to vary the buffer concentrations. So the buffer concentrations are in grams per liter, and now where it gets interesting, we want to use this parameter for the concentration calculation. That means when we are preparing the input mixture for the reaction chambers, we have to add the parameter buffer concentration to the calculation. Also, we have differing buffer types. This parameter has no unit, but we still want to include it in the concentration calculation, so we assign it to the parameter buffer concentration. Now we know how much of each buffer we have to use for preparing the input mixtures. For the parameter pH, we have the same case. It has no unit, so we assign the parameter buffer concentration because the pH of the stock buffer, which we use to create the input mixtures, can be adjusted. For the enzyme, we also have no unit and want to add it to the mixture. But since the enzyme is not in the buffer stock mixture, we have to define the parameter enzyme concentration, to which we can assign the parameter enzyme to. Enzymes are sensitive reagents in the case that high salt concentrations would be destructive for the enzyme. That's why we select sensitive reagents, so when the input mixtures are prepared, the enzyme will be added after the solvent was. Now that we defined all our parameters, we can choose which we want to vary in our experiment or which should be constant. So we can define the values for the constant parameters and can fill the matrix with the values for the dynamic parameters. Okay, now that we planned our experimental setup, we can get to preparing the input mixtures for the plate assay. First of all, we define how concentrated our stock solutions are, so we know if all reagents fit in the specific wells. For example, if we want to run our tests with 20 milligrams per liter enzyme concentration and our enzyme input stock is 20 milligrams per liter, we would need 100% of the enzyme stock to fill the wells, which would left no space for the buffer. If the stock concentration of the pH 6 buffer is low, it only fits in the low buffer concentration wells, but not in the high concentration ones. Only if all of your stock concentrations are high enough and you saved your experimental setup, you can proceed with preparing the input mixtures. And a short addition, you can also modify the labeling method for your matrix and adjust the layout. Okay, now that we checked our stock concentrations and saved the setup, we can proceed with creating the input mixtures for the wells. This is done in multiple steps. Step 1 is preparing your reagent tubes in the order specified in the scheme. You place your reagent tubes with the minimum volume specified for each tube and label them with the number above the minimum volume. In the second step, you add the non-sensitive stock reagents, in our case the buffer. We defined the stock concentrations earlier, so we know how much of each buffer belongs where. The third step is the distribution phase, where you distribute the mixtures from the tubes of the left side of the line to the tubes at the right side of the line. In our case, we could directly put the buffer in the tubes of the right, but if you have multiple non-sensitive reagents, it saves time and pipette tips. In the fourth step, you add the solvent to the marked tubes, and in the fifth step, you add the sensitive reagent, in our case enzyme, to the tubes. Now that we have our input mixtures ready, we can transfer the solutions from the tubes to the wells. The pipetting scheme displays the tubes labeled by the number and where it belongs. If you made a mistake while pipetting, you can change the order of your wells by drag and drop. Saving the changes will also change the metrics, which you can see in the background. Your experiment is saved and you can load it at any time. After you have your results, you can upload them here and transfer your data directly to the database. You have two possible ways to upload your results. First is to upload a CSV file where you specify the well label in the rows with the matrix label, row label, and column label. The columns are named with the Y values you want to add. In the preview, you can see what your results are for which parameters. The second way to upload your data is by filling it in this data matrix, where you can also specify your Y values and upload it to the database with the parameters. Now that we completed our experiment, let's take a look at the results database. Here you see all your previous experiments, including well labels, experimental parameters, and results. For visualization, you can hide columns by clicking on the column header. Also sort the data by parameters with multiple sorting levels, filter the data, and compare your results in a nice plot. By right-clicking a row, you can get to the experimental setup of this experiment, which is stored for data integrity. You can take the setup directly as a template by changing and saving it again, which won't affect the stored experiments from a result table. You can load it again by clicking the button Load Experiment. This is what is included in the basic subscription of the program. If you need more complex processing of your data, for example, you have time-dependent output from the experimental method, you can contact us and we will implement a customized script for you. This could look like the following. Once you created your experiment, you can directly upload your raw data, specify some constraints, and accept or discard the data points. This whole process can be completely customized based on your inquiry. And you can see on the right side of the table some custom Y values, which are automatically calculated. Plus, you have different kinds of data visualizations available. Thanks for watching and enjoy experimenting with the DigitalLabApp.